Hello, sir. So, today I am going to start the presentation for Group 1, which is Biophysical Technique for Ligand Screening and Drug Design. I am doing the introduction part. The biophysical technique for drug screening, and drug design introduction. Ligand based drug design is an approach used in the absence of the receptor 3D information, and it relies on knowledge of molecules that binds to the biological target of interest. To use this, we need at least one active compound. The overall goal is to represent these compounds in such a way that the physiochemical properties most important of their desired interactions are retained, whereas extraneous information not relevant to the interaction is discarded. Ligand-based virtual screening method use the information present to known active ligand rather than the structure of target protein for both lead identification and optimization. Ligand-based methods are only chosen when no 3D structure of the target protein is available. This is typically the case, for example, with G protein coupled receptor targets or protein structures resolved in the apoform. In practice, without knowing the protein structure for a target of interest, one often knowing that a set of ligands are active against the target of interest. As such, ligand based virtual approaches can be employed, that is, finding new ligands by evaluating the similarity between candidate ligands and known active compounds. By using a ligand based method, the known active compounds are collected in order to select a query for virtual screening or alignment in ligand based design. A ligand based computational method involves two essential elements: an efficiency similarity measure and a reliable scoring method. In addition, the computation methods should be able to screen a large number of potential ligands with reasonable accuracy and speed. That's it for the introduction part of the biophysical technique for ligand screening and drug design. Now I would request Millie to proceed with the objectives of this presentation. Thank you.
Hello, I am Mili Deshandra. I am presenting to biophysical technique for ligand screening and drug design. Objectives. Drug design is the innovative process of finding new medication based on the knowledge of a biological target. In the most basic sense, drug design involves the design based on the knowledge of biological target. In the most basic sense, drug design involves the design of molecules that are complementary in shape and charge to molecule target which with their interact and bind. Screening, as the name implies, is a sifting process to find a few compounds in a large set that have desired biological activity. Biophysics represents the perspective that the biological system in question and drug delivery device being developed can be understood through the multi-scale mechanistic paradigm provided by biophysics. The goal of process chemists is to develop synthetic routes that are safe, cost-effective, environmentally friendly, and efficient. The basic premise of screening is that biological assays are reproducible, reliable, and robust, and biologically relevant. Enzo offers a line of product and service that can accelerate your drug discovery program. The aim is to deliver innovative tools that make drug development more efficient, more co-effective, and more successful. Fundamental approaches of the LDCA DDR selection of compound based on chemical similarity to non-active, some similarity measured, or the construction of a model that predicts biological activity for chemical structure. Thank you.
Thank you, Millie. Myself, Krishna Vadher, and I am going to discuss about the methodologies of the ligand-based drug design. In short, LBDD techniques used different methods for describing features of small molecules using computational algorithms that balance efficiency and information content. Molecular descriptors can be structural as well as physicochemical. Examples are molecular weight, geometry, volume, surface areas, ring content, etc. Visual screening is a computational technique used in drug discovery to search libraries of small molecules in order to identify those compounds which are most likely to bind to a drug target, typically a protein. In this project, you will be required to create an algorithm results based on their score so that compounds that score high for the protein of interest do not rank high for the other protein of the same family. It is generated through the knowledge based MM and QM based tools, classified based on the dimensionality of the chemical representation from which they are computed 1D, 2D, 3D, and 4D. Ligand based drug design approaches can be molecular fingerprint and similarity searches, pharmacophore modeling, and qualitative structure activity relationship. In short, QSAR. Similarity searches based methods QSAR and pharmacophore are modeling methods consider physicochemical properties most important for the desired interaction and by discarding extraneous information not relevant to a desired interaction. Let's talk about the molecular fingerprint. Molecular fingerprint based techniques attempt to represent the molecules in such a way to allow rapid structural comparison in an effort to identify structurally similar molecules or to classify collections based on structural similarity. They are less hypothesis drive in and less computationally expensive than pharmacophore mapping or QSLR models. It is a qualitative approach. These consider all parts of the molecule equally and avoid focusing on a part of a molecule that are thought to be the most important for the activity. Let's talk about the pharmacophore modeling. The rationale behind the pharmacophore model is that the inhibitor against a specific target process at a set of common physical or the chemical properties responsible for the mode of binding. These are three processes involved. First is finding the features required for the particular biological activity. Second one is determining the molecular conformation required. And third one is the developing a superposition and alignment rule for the series of the compound. The concept of the pharmacophore has been widely applied to the rational design of novel drugs. Pharmacophores can be used to represent and identify molecules on a 2D or 3D level of the schematically depicting the key elements of a molecule recognition. The most common application of the pharmacophores is visual screening and different strategies are possible depending on the prior knowledge. However, the pharmacophore concept is also useful for ADME, tox modeling, side effect, and target prediction as well as target identification. Pharmacophores are often combined with the molecular docking simulation to improve the virtual screening. The typical types of interaction sites recognized by pharmacophore softwares include hydrogen bond acceptor, hydrogen bond donor, hydrophobic, negative ionic, aromatic rings, and positive ionic. Quantitative structure activity relationship. QSAR is the process of studying a series of molecules of different structure and properties of attempting to find the empirical relationship between structure and properties. As the name suggests, this method correlate molecular structure with biological properties. QSLR methods are based assumption that the structure of molecule must contain the features that are responsible for its physical, chemical, and biological activities. This method is defined as the process that qualitatively correlates structural and molecular properties, which we call as descriptor. It functions like the physicochemical properties, biological activities, and toxicity, etc. QSAR approaching to recollection of act and denied to compound and generate molecular descriptor that describe the physicochemical or the structural properties of the compound and then general the amount and the identifies relationship between the descriptors and the experimental activities. Thank you, and now I would like to pass this opportunity to Drashti Sakaria.
Hello, good morning, sir. I am Drishti Sakaria and I will tell about biophysical technique for ligand screening and drug design result and discussion.
Biophysics represents the perspective that the biological system in question and drug delivery device being development can be understood through the multi-scale mechanistic paradigm provided by biophysics. Ligand-based virtual screening method use the information present the no-active ligand rather than the structure of target protein for both lead identification and optimization. Ligand-based methods are the only chosen when no 3D structure of the target protein is available. It is also important to consider that structure-based drug design direct of the drug lead, which is not a hot drug product but a specifically compound with a least molecular affinity for a target. And molecular docking is the most used structure-based technique, and it applies a scrolling function to estimate the fitness of and ligand against the binding site of the molecular receptor, helping the chosen the ligand with the most high affinity. Computational approaches are useful tools to interpret and guide experiments to expedite the antibiotic drug design process. Structure-based drug design and ligand-based drug design are the two general types of computer-aided drug design approaches in existence. The drug design software such as Insight 2, Material Studio, a Code Prime, and use of structure-based drug design are drug-based drug design such as Glide, Macromodel, Autodoc, and are used for ligand-based drug design. And the most popular approaches for ligand-based drug design are the QSAR method and pharmacophore modeling. QSAR is a computational method is quantify the correlation between the chemical structure of series and compound in the particular chemical or biological process. Pharmacophore is the essential to understand the interaction between the receptor and ligand. It is important feature to design new drug for treatment of the internet disease. The ultimate goal of drug design is the discovery of new chemical entities with desirable pharmacological properties. Achieving the goal requires medicinal chemists to explore the chemical space for a new molecule, which is proved to be extremely difficult mainly due to the size and complexity of the chemical space. A drug target is a molecule in the body, usually a protein that is initially associated with a particular disease process and that could be addressed by a drug to produce a desirable therapeutic effects. Thank you. That's all about my part.
Conclusion of this part is done by me, which is Anisha, because Bharat Bhukia apparently has not responded to any of our texts and comments. So I'll go with the conclusion of physical technique for ligand screening and drug design, which is a ligand based virtual screening method use the information present in known active ligand rather than the structure of a target protein for both lead identification and optimization. Ligand based methods are only chosen when no 3D structure of the large protein is available. The goal of the preclinical drug discovery program is to deliver one or more clinical candidate molecule, each of which has sufficient evidence of biologic activity at a target relevant to a disease as well as sufficient safety and drug-like properties so that it can be entered into human testing. That is all. Thank you for giving us the opportunity to present this presentation. We are signing up as group 1, and with this I will include a quote, which is, with the increase in the technology we should always consider the environment first, because we have to conserve the environment for our future purpose also, and it is our primary goal to conserve it. That's it. That's all, and have a nice day.